Welcome to Bioinformatics.Expert. In this video, I want to continue to show you how you can analyze chip sequencing datasets with the Bioinformatic Tools online program. In the last video, we started with the BVA aligner and I showed you how you can align reads to the human reference genome. However, to show you these functionalities, I used an example FASTQ file which only contained 1 million reads. That means the analysis was finished quite fast. However, when you align the reads of a FASTQ file coming from a real experiment, then the analysis takes much more time. Here you see, for example, a log file of such an alignment. This is created by Bioinformatic Tools Online, and you see that the run took approximately 17,000 seconds, which is actually more than 4.5 hours. Like with the example dataset in the last video, I then used SumTools sort to convert the output sum file to a sorted BUM file. With that, we can now continue with the chip sequencing pipeline. The next step, however, is to remove duplicates with the program Picard mark duplicates. But as it is indicated here, this step is optional, and I already showed you how you can use this tool in the RNA sequencing playlist. So if you want to remove duplicates in your alignment file, then please have a look at the video there. Should you remove the duplicates? This question is hard to answer because the literature is not clear in this regard. However, I think that in chip sequencing experiments, it might be necessary to do this. But as I said, for the sake of this video, which only shows the technical aspects of the analysis, we skip this step here. So what we are now doing is we create browser tracks from the alignment file. To do this, we use the program Deeptools BUM coverage. It calculates the coverage and creates a file which can be used by the UCSC genome browser, for example, to visualize our reads. I will also show you later how you can do that. Okay, but before we start with Deeptools, we have to create an index file of our BUM file because Deeptools only works when the BUM file is indexed. You can easily create an index file with SumTools. I implemented the program SumTools index into the Bioinformatic Tools Online so that you can create this index file with only some clicks. First, click on SumTools index in the table. Then you can select an ID as for all of the tools which are implemented in this program. Then we select the BUM file which we want to index, and then we press continue. In the parameter table, we don't have to add more. We can just press the Start the Analysis button and SumTools will then create an index from our BUM file. With this index, we can now start Deeptools BUM coverage. For that, go back to the Analysis table and press the Deeptools BUM coverage button. Now you can insert an ID or you skip that. And now we select the sorted BUM file from the alignment. Now we have to select the index of the BUM file which we created in the step before. And now we continue to the parameter table. In this case, the parameter table is quite large. However, I separated it into 3 parts for a better overview. The first thing we have to do is to select the output file type. We can select between bedgraph and bigwig. The difference between the two file types is that the bedgraph file can be viewed with any text editor, whereas the bigwig file type is a so-called binary file type. This means it is smaller than the bedgraph file, however, it cannot be read with a normal editor. And when you want to import the file into the UCSC genome browser, then it is much more easier to do it with a bedgraph file than with a bigwig file. For that, we select here bedgraph. The next thing we have to define is the so-called bin size. For example, a 50 here means that the genome is separated into bins of 50 bases, and for these 50 bases, the coverage is calculated. But since we are calculating the coverage of the whole genome, I would suggest to change this value here to 200. The next part in the parameter table is about the normalization. Some of the following fields are necessary and some are not. So if you want to find out which of the fields are necessary before you start the analysis, an easy approach is to just click Start the Analysis button right now. Bioinformatic Tools Online will now give you exclamation marks at the fields which you have to fill out before you start the analysis. For example, we see 2 exclamation marks here. The question, do you want to normalize using RPGC? And the question, if not, please select the normalization method. When we look at the description of the exclamation marks, we see that it says, please provide only a value for one of the marked parameters. This means we have to either fill out this one or this one. If we, for example, select here a normalization method, for example RPKM, then the exclamation mark disappears here. But if we now select that we want to normalize using RPGC here, then more exclamation marks appear. The one in blue tells us that both values are set, but only one is allowed. That means we have to remove this value here, and now the blue one disappears. However, the yellow exclamation mark tells us now that we have to provide all or no values for the marked parameters because they belong together. That means if we want to normalize using RPGC, then we have to add an effective genome size to the parameter table. The effective genome size describes the number of bases in a genome which are accessible by an alignment program, for example. However, there are more definitions around and several sources have several definitions. That means you have to take a look into the literature and find out which definition applies for your experiment. So I would suggest that we don't normalize using RPGC and that we normalize using RPKM. This normalization method does not need any effective genome size. A last word about the exclamation marks. They appear in all tools in the Bioinformatic Tools online program if you forget to fill out a required parameter. If they appear, then please read the description of the exclamation mark, because sometimes it tells you, like here, that you have to fill out either one or the other parameter, or that you have to fill out 2 parameters for the tool to work properly. The final field in this normalization part is this here, where you can select chromosomes which should be ignored during normalization. This field is not mandatory, that means you can fill out it or not. Before we start the analysis now, I just want to show you the last part of the parameter table. This part contains parameters which can be included but which don't need to be included. For example, you can ignore duplicate reads, or you can center reads, or you can remove reads which have a specific SAM flag. However, I would suggest that you click on the question marks if you have a question, because there it is a little bit explained what the parameters do in the analysis. If you then have still questions what that means, then just write me an email and we can clarify it together. Now we start the analysis, and for the whole genome this takes quite long to finish. However, at the end we get this bed graph file with the coverages, and we can then use this file to visualize our results in the UCSC genome browser. To visualize your data now in the UCSC genome browser, the first step is to download this bed graph file. Then visit the web page of the UCSC genome browser, which is https://genome.ucsc.edu. Here, click on Genome Browser. If you are in Europe like me, it will ask you then if you want to stay at the American page or if you want to change for the European page. I click on the European page and it brings me to this web page here. The next thing, which is very important, is that you select now the genome which you have used for aligning your data. In our case, it was the human genome GRCH38, so we select it here and press the Go button. Now we see several information of the human genome, for example, the genes and their names and where they are located within the human genome. And here we can now add our bed graph file. For that, scroll down and look for a button called Add Custom Tracks. Click on it and you will get to an upload page. Here, look for this Browse button and select your Deeptools output file coverage point bed graph. Press Submit, which means it will be uploaded to the UCSC Genome Browser. You then see a table which defines your custom track. But please be aware that the UCSC Genome Browser defines the type of your track to be a bed. And this is wrong. This has to be changed to a bed graph to get proper graphs. So click on User Track and you come back to this page here. In this field here, you have to add the following statement: type equals bed graph. Then you have to browse again for your file and click on Submit so that it will be uploaded again. Unfortunately, there is a bug here, which means you cannot only edit this field here and then press Submit. You have to upload the file again to make it work. We now see that the type has changed to bed graph. This means we can now view it in the Genome Browser. First, you see not so much. This is because it shows you only a very small part of chromosome 1 and the track is also displayed in a dense format. To change that, I would suggest to start by entering here the whole chromosome, for example chromosome 22, and then to change the user track visualization to full. Now you see some peaks here, which you can zoom in by, for example, marking the area and clicking Zoom in. And then you can observe where the peaks are located. Alright, so thank you for watching this video about Deeptools bump coverage in the chip sequencing pipeline, which was performed with the Bioinformatic Tools online program. If you want to use this program in your research or if you want to test it, then write me an email or visit my webpage.